DNA replication. At the end of this module, you will be able to describe the process of replication. DNA replication can be defined as a process in which an entire double strand is copied to produce an identical set of DNA helix. The process of replication starts at a specific point on the DNA. This point is called the origin, or origin of the replication site. The first step in replication is the binding of the enzyme helicase to the origin. The function of helicase enzyme is to unwind and separate the strands of DNA. The strands of DNA are complementary, and the moment they are separated they tend to bind back. This is prevented by proteins called single-strand binding protein, which binds to single strands of DNA and prevent their rebinding. Now, a second enzyme called the DNA polymerase binds to the separated strands and begins the process of replication. But DNA polymerase can add DNA nucleotides only to a pre-existing chain of nucleotides. Therefore replication really begins when an enzyme called RNA primase assembles a small strand of RNA called RNA primer at the origin. The RNA primer is a short stretch of RNA nucleotides which are complementary to a small section of the initial sequence of DNA strand. DNA polymerase is now able to add DNA nucleotides to this primer and initiate the process of building a complementary DNA strand. Once a complementary DNA strand is built, the RNA primer is removed by enzyme action and appropriate DNA nucleotides are added. The two strands of DNA are oriented in opposite directions. But the polymerase enzyme can carry out replication only in one direction. Therefore a different mechanism is used to replicate the opposite strand of DNA. In the second strand moving in the opposite direction, DNA is replicated discontinuously to produce a series of discontinuous DNA fragments. These fragments are called Okazaki fragments. As the Okazaki fragments are built, the RNA primers are replaced with DNA nucleotides enzymatically. The individual Okazaki fragments are then bonded together to form a continuous complementary strand by an enzyme called ligase. The continuously formed strand is called the leading strand, and the strand synthesized in small pieces is called the lagging strand. The process of DNA replication is completed when DNA ligase joins all the short stretches of DNA into a continuous strand.